Earlier methods of pharmaceutical manufacturing relied on end product testing to assume safety. Operators would pull a small number of vials from the end of a manual production line, test them in a lab, and use those results to clear the entire batch for distribution. This approach is statistically flawed. To test enough vials to mathematically guarantee that a batch is completely sterile, you would have to destroy every unit produced, leaving nothing left for the patient. Because of this limitation, the FDA enforces Current Good Manufacturing Practice, or CGMP. These regulations are based on the principle that quality, safety, and efficacy must be designed and built into the manufacturing process from the start, as they cannot be verified through terminal testing alone. This requirement is met through process validation. This is the collection and evaluation of data, from the initial design stage through commercial production, to establish scientific evidence that a process will consistently deliver a quality product. To understand how this evidence is generated, we must examine the microscopic scale of a single unit operation, sterile filtration. By tracking the mathematical validation of a single filter pour, we can see how that logic scales to a full facility lifecycle, and how manufacturers use regulatory tools to adapt those systems over time. Process validation uses data to build a predictive model, ensuring future manufacturing performance by evaluating the relationship between process inputs and product quality. The industry standard for evaluating the efficacy of sterile filters is the ASTM F838 bacterial challenge test. This protocol uses a specific bacterium called Brevundimonas diminuta. B. diminuta is the challenge organism of choice because it is one of the smallest known bacteria, providing a rigorous standard for filter qualification. The baseline requirement for this test is a challenge of 10 to the 7th, or 10 million bacteria, for every single square centimeter of effective filtration area. This creates a 7-log reduction per square centimeter. If the filter is effective, 10 million bacteria impact the membrane, and 0 active bacteria emerge in the filtrate. Filter manufacturers often cite an 11-log reduction in their marketing. That number results from applying the 7-log standard across the entire surface area of a total device, such as a 10-inch pleated cartridge, which contains roughly 7,000 square centimeters of membrane. While the total device may retain over 100 billion bacteria, the physical stress and retention capability on any individual square centimeter remains exactly 7 log. Robust engineering depends on these normalized, specific physical metrics mapped per square centimeter, providing a consistent baseline regardless of the total filter size. There is a practical problem with the ASTM F838 test. Running a bacterial challenge destroys the filter's usability. A filter saturated with live bacteria cannot be used to process a commercial drug. To solve this, engineers use a non-destructive physical proxy, known as the integrity test, to verify the filter without contaminating it. The bubble point test is the primary integrity metric. It begins by completely wetting the filter membrane, which seals every microscopic pore with a liquid film. Gas pressure is applied to the upstream side of the filter. As pressure increases, it overpowers the capillary forces holding the liquid in the pores, tearing the film away and resulting in bulk gas flow to the downstream side. This graph traces the event, plotting gas flow against pressure. The initial shallow curve shows normal gas diffusion through the liquid film. The moment the film tears, the data shows an exponential spike. This transition from diffusive flow to bulk flow defines the specific bubble point of that membrane. In 1979, researchers Johnston and Meltzer established a mathematical link between this physical pressure reading and biological retention. Plotting the biological log reduction value against the physical bubble point reveals a linear correlation. This allows engineers to determine the exact minimum pressure required to guarantee complete bacterial retention. While a manufacturer provides a bubble point for a water-wetted membrane, the drug manufacturer must establish a product-specific integrity value. This accounts for how the specific drug formulation interacts with the membrane during the actual filling process. This correlation allows engineers to monitor an unmeasurable biological attribute by controlling a measurable physical parameter. This logic must scale from a single unit operation to the macromechanics of an entire biomanufacturing facility. This scaling is managed through a regulatory framework that determines how manufacturing processes are approved for public use. Prior to 2011, validation was often treated as a one-time milestone. A manufacturer would run 3 successful commercial batches, archive the records, and consider the process validated. Current FDA guidance describes process validation as a 3-stage life cycle: process design, process qualification, and continued process verification. Stage 1 is process design. Here, engineers use design of experiments at the pilot scale to identify variables and establish the refined control strategy used for commercial production. Stage 2 is process qualification. This stage confirms that the facility, utilities, and equipment are capable of reproducible commercial manufacture under CGMP conditions. Stage 3 is continued process verification. This relies on statistical feedback loops and process analytical technology to monitor production data and identify opportunities for optimization. This lifecycle approach maintains the manufacturing process in a continuous state of statistical control, rather than relying on a historical record of a single success. While a validated state is necessary for safety, it creates a dilemma for the manufacturer. Changing a validated process to improve efficiency or resolve a supply shortage is a complex regulatory task. Significant changes to chemistry, manufacturing, and controls, or CMC, can require months of regulatory review before they can be implemented. Major changes often fall into the Prior Approval Supplement, or PASS, category. These changes require explicit FDA approval of the supporting data before the modified product can be legally distributed. To manage this, manufacturers use a regulatory mechanism called a Comparability Protocol, or CP. A CP is a prospectively written plan submitted to the FDA before a change is needed. It defines the exact risk assessments, analytical tests, and acceptance criteria that will be used to demonstrate that a future change will not adversely affect the product. An approved CP reduces the reporting burden for future changes. It can downgrade a major pass requirement to a CBE 30, which allows a manufacturer to distribute product just 30 days after notifying the FDA. A manufacturer can use a CP to qualify a secondary filter supplier in advance. If the primary supplier fails, the manufacturer executes the agreed-upon tests and pivots to the new source immediately, maintaining the drug supply. A comparability protocol embeds the future evolution of a process directly into its regulatory approval, ensuring that improvements do not disrupt the patient's access to medicine. The architecture of pharmaceutical quality is an integrated system designed to ensure patient safety. It starts at the microscale, where absolute biological limits are used to derive non-destructive physical controls. These controls are integrated into a 3-stage validation lifecycle that ensures continuous statistical oversight of the entire facility. And through comparability protocols, this system is engineered to evolve and scale without compromising safety or supply. When these physical and regulatory systems are integrated, the result is a production line that consistently delivers sterile, effective medicine. Engineering this level of control ensures a process remains effective even as it adapts to new variables throughout the life cycle of the drug.